Welcome to the Immutep TACTI-003 result webinar. My name is Paul Sanger and I will be the moderator for today's session. On the call today, we have Executive Director and CEO Marc Voigt. We have Chief Scientific Officer, Professor Frédéric Triebel, Chief Medical Officer, Dr. Florian Vogl, and Senior Vice President, Strategic Development, Christian Mueller. Questions will be taken from the analysts at the conclusion of the presentation, and copies of the presentation will be available at the Immutep and Finance News Network websites after the session. Without further ado, I'd like to hand over to CEO Marc Voigt. Marc, please go ahead.
Thank you, Paul, and welcome everyone. And I appreciate that all of you are taking the time for the next 40 minutes to go through our results from TACTI-003, also a few aspects of TACTI-004. And of course, an outlook. Paul kindly introduced the management team already, so I don't need to do that. So let's start. So here's my forward-looking statement. And I only have a few initial slides just to set the background for those who don't know us that well. But I promise not to spend too much time there. So here you see our different programs. We will clearly focus on eftilagimod alpha, the program you see on the left-hand side, a unique antigen-presenting cell activator, where we have a unique way to activate the immune system. On the next slide, you can see our pipeline in oncology and in autoimmune diseases. We are very excited about the progress we are seeing. For instance, the bottom was IMP761, where we are planning to start a first-in-human trial mid of this year. Very excited about the progress in first-line non-small cell lung cancer. And of course, and this will be the key topic of today, about the progress in first-line head and neck cancer. We run these programs in a collaboration with Merck, where we have actually 3 clinical trials, TACTI-002, TACTI-003, and TACTI-004, with such a strong partner to combine Keytruda with eftilagimod. On the next slide, here we see eftilagimod again, and we see how it's uniquely working in the crosstalk of innate and adaptive immunity. And we see efficacy across hot, tepid, and cold tumors in patients with high, low, or negative PD-L1 expression. And this is what we saw in the past, and yet again. On the next slide, you see also that we are addressing large markets. Non-small cell lung cancer clearly leading. We are also active, often overlooked, in metastatic breast cancer, and of course, exciting in head and neck cancer. And now I would like to hand over to Dr. Florian Vogl, our Chief Medical Officer. Florian, go ahead.
Thank you, Marc. So when we talk about head and neck cancer, we really talk about cancers that arise from multiple locations of the head and neck, as you can see on the image here. And they are very different entities, making this a difficult to treat heterogeneous disease. In this trial, TACTI-003, we included cancers that originated in the mouth, the throat, and the voice box, here shown in red, which all fulfill essential functions like swallowing, talking, breathing. What these cancers have in common is they form in the lining of the structures known as the mucosa and are referred to as squamous cell carcinoma. Squamous cell carcinoma makes up for approximately 9% of head and neck cancers. Well-known risk factors for head and neck squamous cell carcinoma include tobacco and alcohol use, and in particular for cancer of the oropharynx, the middle part of the throat, infection with the human papillomavirus, HPV. From an epidemiology perspective, there are approximately 890,000 new cases diagnosed every year globally, and it is estimated that approximately 100,000 patients in the major markets develop recurrent or metastatic cancer, which means that the disease returns after initial treatment or spreads to other organs. The 5-year survival rate of metastatic head and neck cancer is around 40% and depends on the anatomical site where the cancer originated, with some more difficult than others. Overall, unfortunately, it's a very aggressive disease. So on the next slide, we talk about the current standard of care, standard treatment. The median overall survival of patients treated first line of metastatic or recurrent head and neck cancer. Taking the best standard of care is approximately 1 year, and there's a high unmet medical need for improvement of treatments that are both tolerable and efficacious. For the treatment decision, patients are classified by their PD-L1 status. PD-L1 is a protein that helps regulate the immune system, as you can see on the right-hand side of this slide. Generally, the high PD-L1 expressors respond best to anti-PD-1 therapy, like Keytruda. Low respond suboptimally, and negative have negligible responses. Typically, the patients whose CPS is positive will receive Keytruda plus doublet chemotherapy or Keytruda alone without chemotherapy. However, if the CPS is negative, meaning the CPS is less than 1, patients will receive chemotherapy plus cetuximab. Note that these patients have no chemotherapy-free options available to them in the first line of treatment. Moving now to slide 10, you see the design of our TACTI-003 study. This is a randomized multicenter phase 2b trial to test FD in combination with Keytruda versus Keytruda alone in first-line head and neck squamous cell carcinoma. Patients were included if they were at least 18 years of age, had recurrent or metastatic disease that was deemed not curable with local systemic therapies, and for which patients had not received treatment. Patients also must have had measurable disease and ECOG performance status of 0 or 1. This study was conducted in 29 clinical sites across many countries, including the US, UK, and countries in Europe. We enrolled a total of 171 patients. Patients with any PD-L1 expression were assigned to cohort A and then randomized to receive either FD in combination with Keytruda or Keytruda alone. Patients with negative PD-L1 expression were assigned to cohort B and received FD in combination with Keytruda. Now, these patients could not be randomized because Keytruda is not approved as monotherapy in this setting. The primary objective of the trial is to evaluate the overall response rate among patients evaluable, which means they had at least one post-baseline scan and were evaluated according to RECIST 1.1. Secondary objectives are looking at further outcome parameters like overall survival and progression-free survival. Turning to slide 11, you will see that 149 patients of the 171 enrolled were evaluable for the primary endpoint. Thus, the sample size of evaluable patients in study cohorts are shown on this slide. In cohort A, there were 58 patients in the combination arm, 60 patients in the monotherapy arm, and then 31 patients with negative PD-L1 cohort B. Patients were recruited irrespective of the HPV status and PD-L1 expression. The level of PD-L1 measured as CPS was used for the assignment of patients to either the randomized cohort A or the non-randomized cohort B. As we speak, many patients are still on treatment and data collection continues for additional analysis, and we look forward to providing more data at the conference in the second half of this year. Let's now go a bit more into important details. There are several prognostic factors well established for head and neck cancer. As depicted on this table, some of the factors have a positive impact on treatment outcome, and you can see those listed in the green column on the left, while others have a negative impact, which you can see in blue color. In the randomized cohort A, we have observed several imbalances between the treatment groups, with multiple prognostic factors clearly favoring the Keytruda monotherapy arm. These include gender, smoking status, the site of the primary tumor, and a greater than 2-to-1 difference in the HPV positive status. Let's talk about safety here. We are pleased to report that there are no new safety signals for FD in combination with Keytruda across cohort A and cohort B. Additionally, the treatment-related discontinuation rate was low and comparable between treatment regimens. This was the first time that FD was combined with a 40 milligram dose of Keytruda, and this favorable outcome, while it's not unexpected, was pleasing to see. Furthermore, there were no treatment-related deaths in the study. While we did see local injection site reactions in patients who received FD, this was expected and were of low grade. We will show more detailed safety data at a conference later this year. And now let's move on to the efficacy data. We are very excited that FD in combination with Keytruda achieved a 34% overall response rate, regardless of the HPV status and PD-L1 levels. And this includes patients with negative PD-L1 expression, where, as we said, the anti-PD-1 monotherapy has shown negligible effect. Consistent response rates above 30% across high, low, and negative patients offers evidence of this novel IO combination's ability to improve patient outcomes and to expand the patient population that benefit from anti-PD-1 therapy. This point was emphasized by Dr. Martin Forster, clinical investigator in TACTI-003. The ability of FD to potentially expand the reach of anti-PD-1 therapies to the point where they are agnostic to the level of PD-L1 expression is a powerful concept. And we are now in the enviable position of seeing this across multiple cancer types, including head and neck, non-small cell lung cancer, and melanoma. Additionally, FD plus Keytruda led to higher objective response rates across all levels of PD-L1 expression versus Keytruda monotherapy in the TACTI-003 trial, despite these treatment imbalances that I already highlighted. And furthermore, we are pleased to see the quality and durability of responses tracking well, and we look forward to sharing more on this front at a medical conference later this year. On the next slide, we're looking at the overall response rates in the randomized cohort A by level of PD-L1 expression. So let me walk you through this table. In patients with high PD-L1 expression, the ORR was 31% for the combination versus 18.5% in patients treated with Keytruda alone. This is a 68% relative increase and a 12.5% absolute increase with the addition of FD over Keytruda alone. Note that this is the largest patient population by PD-L1, representing approximately 50% of first-line head and neck patients. In patients with low PD-L1 expression, FD plus Keytruda has a relatively high overall response rate of 34.5%. With Keytruda monotherapy, the objective response rate was 33.3%, which is higher than the historical published data, including an ORR of 14.5% in this group of patients in the KEYNOTE-48 trial. The relatively high ORR in TACTI-003 in this patient group may be explained by the imbalances in markers of prognosis that I already talked about, specifically gender, smoking status, HPV status, and the location of the primary tumor. And as a reminder, all of these were in favor of the Keytruda monotherapy arm. Now, at the bottom is shown taking these two subgroups together as the CPS greater equal 1, positive PD-L1 expression, FD in combination with Keytruda has an ORR of 32.8%, and Keytruda alone a 26.7% response. This corresponds to a 23% relative increase over Keytruda monotherapy, whose ORR was driven higher in part by the unusual results in the CPS 1 to 19 group. The difference between the treatment arms in cohort A was not statistically significant, as reflected in the confidence interval, and this is due to the small sample size of evaluable patients combined with the imbalances favoring Keytruda monotherapy and the very large differential required. Having said that, we are very happy that FDM Keytruda has driven a consistently high overall response rate of roughly 34% across all levels of PD-L1 expression, including negative expressing patients. So a word about cohort B. We're now on slide 16. We have previously reported a preliminary ORR of 26.9% and a disease control rate, DCR, of 57.7% for patients treated with FD in combination with Keytruda. These are the patients who do not express any PD-L1. As shown in the graphic on the right, these results compare favorably to the historical 5.4% ORR and 32.4% DCR in evaluable patients from KEYNOTE-48 study in the same patient population. Today, we can share that the ORR in this CPS negative group in TACTI-003 has substantially improved over what we previously reported. The exact results will be presented at an ESMO virtual plenary session on July 11th. And turning to slide 17, you can see that ESMO virtual plenaries are a great opportunity to present original scientific data from phase 2 trials, which are selected if they demonstrate remarkable therapeutic benefit, scientific insight, or progress in an area of unmet need. And probably we met all three criteria. Anyway, we are honored to have been chosen, and we're excited to provide the latest clinical data from FD plus Keytruda in this difficult to treat patient population who really have no effective chemotherapy-free treatment options. And with this, I'd like to hand over to Christian Mueller. Christian?
Thanks, Florian. Let me start with a summary of what we just heard, just that we maybe recap the highlights from what Florian just explained. We saw a consistently high response rate, above 30% for FD plus pembrolizumab, regardless of PD-L1 expression. And this includes patients with a negative PD-L1 status, as Florian just explained, and we will highlight that definitely. Or we will present more on that during the ESMO plenary. Most importantly, this is in line what we have seen in other indications like non-small cell lung cancer and melanoma. So it gives us further support on the data. I would also like to highlight the strong performance in the randomized setting in patients with high PD-L1 expression, where we saw the relative outperformance of about 68% increase in ORR compared to Keytruda alone. So what's next? We look forward to provide an update, as Florian just said, at the ESMO plenary in a few weeks, and this should be really exciting given the data we will have. And beyond that, we will share more information during a medical conference in the second half, which actually is in a few days from now. And besides that, based on these positive top-line results we just saw, we will discuss the path forward in first-line head and neck with regulators, including FDA, where we have the fast track designation, and we will focus obviously on areas with a high unmet need, as this whole field of head and neck is. Now I would like to shift gears and focus on our lead indication, first-line non-small cell lung cancer. We have been fortunate, as depicted here on the pictures, to receive different oral presentations at major medical conferences, capturing the evolution of our non-small cell lung cancer data. This started with a high overall response rate initially, then evolved into strong progression-free survival, durability of responses, and ended last year with excellent overall survival. And based on this pretty strong data set, we embarked on a new collaboration agreement with MSD or Merck to supply Keytruda for a phase 3 registrational trial in non-small cell lung cancer. The value of this collaboration is significant to us in several different ways. As Keytruda, the supply alone of Keytruda is worth approximately $100 million for a trial of that size. And as Keytruda is the #1 brand in the PD-1 field, it will give us access to a huge market in case the study is successful. Most importantly, we retain all commercial rights of FD, which will give us full flexibility in terms of business development. We believe FD and the study are uniquely positioned for trial success, as we have been discussing the study itself with all the important stakeholders, and all of them have been bought into the concept. This basically means MSD, regulators across the world, PIs, investigators, and other stakeholders. And I believe one of the key factors for trial success, especially in terms of recruitment in such a high competitive environment, is that we are not coming with a me-too drug, but with a unique concept in terms of an agonist, and also here with a quite excellent safety profile, as Florian just explained a few seconds ago. And we are very confident in our positioning here, thanks to the strength of the mature data from over 130 patients, and it includes mature PFS and mature OS, which are the usual endpoints for phase 3, which will make the translation hopefully a lot easier into that registrational setting. So we believe the trial design, I will show you in a second, is uniquely set up for success in a really, really large market opportunity. So let me walk you through the important aspects of the trial design, and honestly happy to answer questions later on. At the top slide, you can see the graphic depicting the design of this registrational study. It's a large 750 patient study, randomized, global, and it will include both squamous and non-squamous patients and cover all levels of PD-L1, so 0 to 100%. Collectively, this represents the largest target patient population you can have in first-line non-small cell lung cancer. FD will be given on top of double chemotherapy plus pembrolizumab versus the same backbone and the placebo. What we have discussed and agreed upon is to have a dual primary endpoint of PFS and OS, which will allow us to look for early success in terms of PFS, but do not compromise anything on the main endpoint overall survival. The trial itself will be formally positive if either of the two is successful. And I think it's not necessary to say, but I'm doing it anyway. The study is obviously adequately powered with that sample size for both dual primary endpoints. And just to reassure everyone, study start preparations are ongoing as we speak. Moving to slide number 22, I would like to explain how we see the unique positioning in the first-line non-small cell lung cancer field. TACTI-004 is one of the few global phase 3 studies in that indication that addresses the entire PD-L1 landscape, so from 0 to 100%. Usually, like anti-TIGITs, mostly they focus on TPS above equal 50, and then you have other concepts. They're maybe focusing on TPS less than 50, but there are not a lot of phase 3s ongoing with 0 to 100%, and then including both squamous and non-squamous patients. And most importantly, we have Keytruda in both treatment arms, so the #1 drug here in that market. As 7 to 8 out of every 10 metastatic lung cancer patients are even still treated today with Keytruda, despite some other approvals. As a result of that high market penetration, it is estimated roughly 36% of the 25 billion sales of Keytruda last year were coming from lung cancer. So clearly, if TACTI-004 is successful, it will allow FD to become part of a massive multi-billion-dollar opportunity and potentially drive a new standard of care for vast majority of patients in the first-line lung setting of non-small cell lung cancer. So all in all, excellent study design built on strong data with huge market addressed. And with that said, I will now turn back to Marc for closing comments. Marc, floor is yours.
Yeah, thank you, Christian. Thank you, Florian. I believe it's exciting what we heard. And of course, we will hear more in the second half of this year, and also, of course, in the next year. We have a very strong, very robust fundament in terms of data, in terms of financial resources. We are looking very much forward to turn this company into a full-fledged phase 3 company. It's happening as we speak, practically. We are very excited that we have already the next stop in terms of data at 11th of July. It's an ESMO plenary session where you can expect quite a big podium for us. It's a virtual plenary session, so no nice picture, but still it will be very nice. It's a very, very important forum. And of course, regarding cohort A and cohort B, a lot of additional data and more details will follow. So we plan also, of course, to go to additional medical conferences in second half of this year, as well as next year. We will see updates from non-small cell lung cancer, soft tissue sarcoma, metastatic breast cancer, and so on and so forth. But I should stop here so that we have also enough time for a Q&A. So thank you so much, and I will hand back to Paul now.
Thank you, Marc. So as Marc said, we will now take some questions from the analysts. You can click on the raise the hand button at the bottom of your screen to enter the queue. And please, in the interest of time, please try to restrict yourselves to one question only. There's a number of hands raised already. Our first question today comes from Eloise Shapiro from Canaccord. Elsie, please go ahead.
Hi, guys. Congrats on the data, and thanks for taking the question. When we look at some of those baseline characteristic imbalances in the cohort A, are you able to provide more color on those imbalances by CPS subgroup?
Yeah, thank you, Elise. That detail of data is not part here of the slide deck, but it's reasonable to assume that you see imbalances across the board, and maybe stronger, especially in the 1 to 19 CPS group. The imbalances as such can happen in smaller randomized clinical trials. This is why you typically do phase 3 clinical trials with hundreds and hundreds of patients so that it balances out. In smaller randomized trials, this can happen. It's an artifact. I don't know if my colleagues, Florian, Christian, Frédéric, would like to add to that. If not, we can go maybe. Yeah, Florian, please.
No, just to underline what you just said, this is we're dealing with very small groups then, because we're slicing and dicing data, but I agree that it's worth looking into it.
Okay, so you can confirm that the low expressors then had more favorable baseline characteristics than the high expressors?
This is definitely reasonable to assume, yes. And this is practically across the board.
Got it, thanks. And any p-values as well?
No p-values, especially not for the subgroups. You see the overlap of the confidence interval. So obviously, the results are not statistically significant. We have here an outlier from my perspective in terms of the 1 to 19 Keytruda group. I'm not aware of any other historic results which showing 33% overall response rate in that group. We have overall a relatively small sample size. And as you may recall, we have been, when we started the trial, setting an ambitious goal. And this ambitious goal for stellar results, which would have been meeting statistical significance given the sample size, and I believe different analysts have been looking into that. It actually mapped out the amount of difference requiring then several hundred patients, like for a traditional phase 3. So the differences you would need to see either in the subgroup or overall would need to be very large. So a 70% difference, which we see almost in greater equal 20, wouldn't do the job.
Great, thanks.
Thanks for your question. Next question comes from Madeleine Williams at Wilson's. Madeleine, over to you.
Hi team, thanks for taking my questions. My question's really focused around sort of going off on what Elise was asking. I mean, your thoughts, and I know it's probably early, but your thoughts in regards to how that will be viewed from the FDA and other regulatory parties for accelerated approval, and whether that high response rate in the Keytruda monotherapy arm is sort of potentially a cause for concern?
I wouldn't, thank you for the question. I wouldn't call it a cause of concern. This is something which needs to be analyzed based on the data. I believe we provided a number of different factors which may explain those results. Definitely, if you look at the 3 segments, and we deliberately designed the study to have different shots on goal. And we can conclude already for CPS negative as well as for CPS greater equal 20, we see a signal in terms of overall response rate. We also should not forget that overall we need to see, of course, follow-up endpoints like progression-free survival, duration of response, and then finally overall survival. Especially as you see in head and neck cancer, not always a straight line. There are different clinical trials you could look at, like the LEAP 10 clinical trial, which had actually statistically significantly positive results in terms of overall response rate, but then a failure in terms of overall survival. So we will definitely discuss with the regulators. There could be some very nice sweet spots. And I believe these sweet spots will also be clearer when we discuss more details in upcoming conferences.
Thank you, that's helpful. I'll go back in the queue.
Thanks. Thanks for the question, Madeleine. Next question comes from Andrew Payne at CLSA. Andrew, over to you.
Yeah, hi. So just following on from that comment from you, Marc, on the LEAP study. So obviously for us, the strength of Keytruda mono wasn't a surprise given the high response in the LEAP study versus Keynote. But can you just dig into that LEAP study a bit more, just in terms of the response rates, progression-free survival, and overall survival data?
Yeah, thank you, Andrew. And I believe that's actually a very interesting case. So the LEAP clinical trial, which has been done by Merck, our partner Merck, in collaboration with Eisai, another large pharma company. So they tested Keytruda plus the Eisai drug versus Keytruda in a phase 3 clinical trial. And this clinical trial has been delivering statistically significant results in terms of overall response rate. So in the high 40s versus mid 20s. And has also been a hit in terms of progression-free survival. But unfortunately for the patients, the overall survival was then finally stronger in the Keytruda mono arm compared to the combination arm. So you see that there is not always a straight line in terms of the efficacy parameters. We do see here with the eftilagimod results, superior overall response rate across the board with a very tight margin in 1 to 19. We did see also, for instance, in other indications, strong progression-free survival, strong duration of response, something which we saw, for instance, in second-line head and neck cancer as well as then finally strong overall survival. So eftilagimod typically transferred across the board of different efficacy parameters.
Marc, maybe it's worthwhile to remind this Christian speaking. Maybe it's worthwhile to remind everyone that we talk different mechanisms of action here. So in the LEAP study, it was a TACTI, so meaning a small molecule inhibiting some transduction signals, typically leading to responses, but they are short-lasting. Whereas if you give IO therapy alone, this is what we are doing, having an APC activator together with an anti-PD-1. If you have a response, you typically see that they are lasting very long, as with an anti-PD-1 alone. So the beauty of seeing an increase in response rate with FD plus pembo is it usually translates into a longer survival for these particular patients, as they have a very, very long-lasting response. So just if you take the second-line head and neck or first-line lung as an example, you see duration of response for more than a year or almost 2 years. Whereas, especially here for second and first line head and neck, the median OS is around about 12 months usually. So meaning these increase in responses we see here for across the board are really meaningful. And this is different to other drugs where you have an impact on the durability of the response. So it's very important to keep that in mind. And because you discussed the monotherapy activity of pembrolizumab in the lead study, I think what is unknown for now, at least we couldn't find it, is the difference for the different CPS subgroups and how many patients have been CPS high and versus CPS 1 to 19 and so on and so forth, and the baseline disease characteristics. I haven't seen anything on that, and that definitely plays a role, especially in such a heterogeneous disease like head and neck.
Thank you, Christian.
Thanks for that question. The next question comes from Dave Stanton at Jefferies. Dave, over to you.
Morning, team, and thanks very much for taking my questions. Look, given this data in first line head and neck, which subsector of the market does Immutep have as its primary potential target market? Thanks.
Thank you, Dave. That's a very interesting question. So the results suggest, in principle, first of all, I put the 1 to 19 aside, suggest a signal in CPS below 1. Let's see also what we have to say 11th of July, and a CPS greater equal 20. And then there are definitely different ways how you could move forward. We discussed in CPS below 1 that there are no chemo-free options available to the patients, and the number of patients don't want to take chemo or can't tolerate chemo. So there could be an option, could also be an option with chemotherapy in CPS below 1. Same applies to CPS greater than 20. But we will simply follow the data and also the discussions. Together, these two segments represent about 70% of the entire head and neck cancer patient population measured by CPS score.
Thank you.
Thanks for the question. Next question comes from John Hester at Bell Potter. Sorry, over to you, John.
Hi, I might just jump in. This is Thomas Wakeham from Bell Potter. Can you hear me all right?
Yeah, please go ahead, Thomas.
Yep, thanks all, and thanks for the presentation today. First question on overall survival, can we expect 12-month OS percentage data, perhaps from the cohort A, towards the second half or early of next year? And then a second question, if I may, is actually on safety. You put some discontinuation data there on one of the slides. Is it possible to get an understanding of what that discontinuation data is only for cohort A, so the FD arm compared to Keytruda, just for cohort A? Thank you.
Yeah, first of all, Thomas, indeed, there will be, of course, overall survival follow-ups, duration of response, PFS. Typically, duration of response has been strong with eftilagimod, also overall survival. So the later we are in a position to update, if it is a landmark analysis or a median or both, remains to be seen. Could be indeed more likely early next year. Some other parameters could maybe also be earlier. And in terms of the discontinuation rate, which is about the same between the arms, which is in line with what we saw in second-line head and neck cancer and first-line non-small cell lung cancer, is related to cohort A, but happy to be corrected by Florian if I made a mistake here.
No, I think so. Your question was twofold, what the discontinuation really means. And let me just explain that the discontinuation here is due to adverse events that are considered treatment related. Now, to do so, we grouped patients together from cohort A and cohort B, all patients who received Keytruda in combination with FD. We don't see a reason why they should be different based on their CPS score versus the cohort B. That is what we've so far done, and that's what was on the slide.
Okay, thank you for that.
Thanks for that question. Next question comes from Joel Beatty at Baird. Joel, over to you.
Great, thanks for the presentation today. Any thoughts on what the ORR findings presented today could mean for the survival data? For example, with the ORR benefit being mainly in the high PD-L1 expressors compared to the low, would that suggest that maybe a survival benefit would be seen in the high PD-L1 expressors, but not the low?
Good question, Joel. Indeed, in case we see a same pattern like we saw in the past, so the translation of the durability, so the quality of the responses, then that should be also reflected in an overall survival benefit. Currently, duration of response tracks well, which is and has been always an important aspect for us. So typically, you do see translation of a good quality of the response into overall survival. So this is an aspect which is quite important to us, and the chance is definitely there.
Thank you.
Thanks for that question. Next question comes from Ahu Demir from Landenberg. Ahu, over to you.
Thank you for taking my question. Good morning and good evening to everyone. I have twofold questions. So I think it's the follow-up from many analysts that asked the question. So in the development of FT across multiple indications, have you observed multiple examples where you see the modest ORR translating into an improved OS, that we don't see major differences between low and high, and based on our analysis, that has not been the case in the lung cancer, and I understand that's head and neck and the context is different, but just curious to hear how frequently have you observed this? Are there any cases, and what was the data in the second line of head and neck that could be maybe more relevant?
Yeah, in terms of second-line head and neck cancer, the median duration of response has not been met. We saw there a different overall response rate pattern. So we saw nothing major really in the CPS below 1. So few patients also in that clinical trials. Overall, if I recall correctly, 36 patients. We saw also in the 1 to 19 only a very modest overall response rate, which was about 10% or so overall response rate. And the TACTI-002 second-line head and neck cancer trial, we saw a very high overall response rate in the TPS greater equal 20 back then. But overall, we had good results. So you see also there, even with modest overall response rate back then in the 1 to 19, we saw about a 50% increase of overall survival in CPS greater equal 1. But maybe also Christian can comment on some of the other studies and the aspects. And one should not forget to put it into historic context. If our data would have been, this is of course a very speculative statement, would have been along the lines what you would historically expect, then of course the question of statistical significance would be different, A. And B, typically you saw in CPS above 1, 19% overall response rate. And we see consistently above 30%, which indicates that the FD results are not an outlier, but consistently reach above the bar. And yeah, Christian, maybe you have additional comments here.
Yeah, first of all, I think the what you look at in terms of PD-L1 is slightly different between lung and head and neck. So it's TPS for lung and CPS for head and neck. So different types of cells you look at. And so what we have seen across the board is that ORR, if induced by FD plus pembro, translated into, because of the good duration of response, translated for those patients into excellent overall survival. This is not only true for NSCLC, this was true for second-line PDX refractory NSCLC, second-line head and neck, and also in the melanoma study we did early on. Another pattern we have seen across different indications is that we see activity in PD-L1 negative patients. So in the TACTI-002 lung cancer, we saw, so this basically PD-L1 negatives are what I would call primarily resistant to PD-1, right? So we have seen a 30% response rate in non-small cell lung cancer there. We have seen now here above 30% response rate in head and neck, where you would expect around 5% from Keytruda alone. And given the sample size we have, one will see that the confidence intervals look pretty good. And so this gives us really confidence that the signal we see in the CPS negatives across the board is really something, and it's what you would actually expect, right, from an APC activator. So it fits to the mechanism of action. And I think other than that, Marc has made all the necessary comments. Maybe you can, if we have missed to answer some parts, maybe you just happy to, give it a second shot.
No, I think you answered my question perfectly. If I could squeeze one more question, I think the remaining question would be, you have a larger market opportunity with lung cancer. Would you pursue this in the pivotal setting, or would you partner this? What would be the strategy that you would pursue?
Yeah, first of all, indeed, we have the phase 3 in first and non-small cell lung cancer, which looking at the market, looking at the data, looking at the collaboration and the opportunity is clearly our favorite. This is what we will concentrate on. We will have definitely regarding head and neck cancer and subject to data from metastatic breast cancer also there. Discussions with the agencies regarding our regulatory path and development path forward in general. So the data which we saw today warrants those discussions. And then we need to make the adequate decisions at the adequate point of time. So I can't give you at this point of time, especially as one should look at the data closely, one should see some more mature data here and there. Then we will make the adequate decisions, and partnering is, of course, always an option.
Great, thank you.
Thanks, everybody. That's all we have time for today. If there are any further questions, please direct them to Marc and his management team in their endeavour to answer them. I'd like to hand back to Marc to make concluding remarks. Back to you, Marc.
This will be very short. I would like to thank everyone on behalf of the Immutep team. As a reminder, this webcast will be available for replay shortly on our website. And thank you very much. Goodbye.